The second helpful activity in Chapter 10 is to help you understand the process of protein synthesis. There are two steps to protein synthesis: transcription and translation. To help you understand that, we've got more beads and pipe cleaners activities. At the start of this one, I once again give you a strand of DNA. First thing you want to do is string the beads in the correct order, and they should basically just look like that. Okay, this strand that you guys just built, that's a gene for a particular thing that we want to make. Like there's a gene for insulin in your body. This is just one of those genes. The first step for us to make the particular protein product like insulin is to carry out transcription. And transcription uses the same basic process of complementary base pairing. It's just that in the process we're going to be making an RNA molecule instead of DNA, and RNA does not use thymine. So if on the original strand you have an adenine, you would have put a thymine there if you were building DNA, but you're not. You're building RNA instead, so you're going to put a uracil as the base pair to adenine. If there's a thymine on the original DNA, it's still going to base pair with adenine on RNA, and the G's and C's go together just like they did before. The only really tricky part to this is that there's no thymine finished on the RNA. When you're done with transcription, you have this strand of RNA, which would be known as messenger or mRNA. The second step in protein synthesis is known as translation, and in translation, the ribosomes read a molecule of messenger RNA, and then they translate that into the actual protein, again like insulin if that was what we were trying to make. So if you look at your strand on RNA, you're going to read it 3 letters at a time, and those 3 letters that correspond to a triplet on DNA, they're called a codon. So the first codon in the example that you had was AUG. You have to have the genetic code in order to carry out translation, and what you do is you look for that exact codon in the box, and AUG is right here. It tells you that that codes for the amino acid methionine, or just MET is the 3-letter abbreviation for that. Then you move down to the second codon, which is CUC. You find that on the genetic code, and that codes for leucine. And then you just keep going until you get to the end. By the time you're done, you have this protein product, which again, this could be an insulin molecule to help you lower your blood sugar, or it could be growth hormone to help a person get bigger. But in order for you to make those protein hormones, you have to carry out transcription to make the messenger RNA, and then translation to make the actual protein.